we go to Dr. GAGAN DUDEJA, D08805, free paper 2175, epithelial to mesenchymal transition in retinoblastoma tumors, a new target. GAGAN, please.
May I start, sir?
Yes, please.
Good afternoon, everyone. Respected chairperson, dear friends. I'd like to present our work to you on epithelial to mesenchymal transition in retinoblastoma, a new intervention target. I have no financial interest to disclose. So just a bit of background on this study. My slides are stuck. Technical help. Yeah. So retinoblastoma is one of the few diseases which come across in ophthalmology where life is at risk. And we all know India has got largest number of new retinoblastoma cases every year in the world. And most of our cases, when they come to us, are something on our right. They are advanced DEs and fairly, even worse than that. So what is causing this? There are multiple factors. Whereas in the West, if you see, we have more of early diseases, ABC, and with current treatment modalities, alone or in combination, there is a very good eye salvage and life salvage rate. Given our scenario, since West is not seeing these kind of patients, they are not going to do any research on that. So once there was an option of doing something, I figured out that this group is one which needs our attention, and maybe we can do something for our Indian cohort and make some difference in life of these patients. So once we reviewed the literature, we find that EMT is responsible for drug resistance and metastasis in many other cancers. Its role in RBU is not reported. We thought, let us check if it can be at play in retinoblastoma. So just to explain what EMT is, epithelial cells, they lose their cell polarity and cell-to-cell adhesion. They will become loose, and then they become mesenchymal, transform into mesenchymal cells, and they can migrate and then transform back. So in the cancer, this will cause, initiation of cancer, invasiveness of cancer. It can also lead to metastasis, as in this figure, and it also causes resistance to chemo and radiotherapy because at that time they will transform again. In the literature, we already have epithelial markers known, which are onto your left, and we have mesenchymal marker knowns, which are onto your right. So going with this knowledge, we devised an experiment that will look in for EMT markers in retinoblastoma tumor, develop an in vitro model to see and understand what signaling mechanisms are associated with it, and see if it can function or help us some way clinically. So the first step was we took enucleated eyes of retinoblastoma patient and checked for gene expression on microarray. If you see here, all these markers are upregulated, and these ones are the one which I showed you before. And down what you see are the genes which are down-regulated. Again, I showed you before. So it confirms that EMT is at play. But our interesting finding are these two, ZEB1 and ABCB1, also known as MDR1, multi-drug resistant gene 1. So we found that ZEB1 and ABCD1 were also at play. So this is an interesting finding in addition to what is reported in literature. So we also want to check for proteins from this thing. And so this is a staining. And if you see here, ZEB1 and CTSL, that is cathepsin L, when we merge them, you can see that they are present at the nuclei of advanced retinoblastoma tumor, but they are kind of absent here in non-advanced and in control. So there's no ZEB1 in control retina. And I'll explain this a little later also. So after doing this thing, these were enucleated cell eyes. To experiment, we need live cells. So we have taken VERI1 RB1 cell line, and then we again did a qPCR to check for the same findings. So here, if you see NCAD and Vimentin are high, which are associated with advanced stage of tumor, these blue bars. And then we also find high FN1, SMA, twist, and snail, which are associated with tumor metastasis, these blue bars here. And there was a high BCL, XL, and AKT, which are associated with better survival and chemo resistance. So the green ones are the control. All the blue bars which are high means they are high. So this is again a proof of EMT markers being there and EMT at being play. But this figure, this small one on the corner, is the surprise. So we also have chemotherapy resistant cell lines. So topotecan resistant cell line and carboplatin resistant cell line, which we have developed. And here we see ZEB1 is elevated. So we find that ZEB1 is there in cases of chemo resistance. So next step is to validate this. We need to check for proteins from these genes. And if you see here in the Western blot, Vimentin here, twist here, and N-cadherin are raised. These are again markers of mesenchymal markers. E-cadherin is low, which is epithelial marker. And interestingly, you also find phosphobeta-catenin raised. Phosphobeta-catenin raised is an indicator of Wnt pathway. So the earlier slide where I showed you about ZEB1, this is a schematic of that. So we know here now from our experiments that ZEB1 activates EMT or epithelial mesenchymal transition in a retinoblastoma cell. And then it goes through further pathway through high N-cadherin, and finally develops a high cathepsin L, which was the green and red slide which I showed you. This high cathepsin L is responsible for tumor invading blood vessels and then, extravasating, causing micrometastasis and metastasis. So this is a probable hypothesis where ZEB1 and cathepsin L are contributing to metastasis. This slide is the best or most interesting slide of this entire presentation. This is where I get excited. We already have drugs which can target EMT pathway in other cancers, and they are being already used. So I'll just stop for a minute and you can see through the drug list on your left-hand side, and they are available. So now, coming with this knowledge, I would just conclude that EMT associated with tumor metastasis in drug cancer is what we know. What our study shows is that first time we are finding the role of ZEB1 and MDR1 resistant gene in EMT and also in drug resistance in retinoblastoma tumors. We also find that if we suppress EMT, we can halt metastatic propensity and reverse chemoresistance in retinoblastoma, which fills in a gap in our knowledge regarding why some tumors are chemoresistant and why some tumors metastasize more and more aggressive. It also provides us a new therapeutic intervention target, which is the most interesting thing. We have shown you the list of drugs which are available. And it also shows that ZEB1 transcription factor and Wnt signaling pathway are at play. So they also give us another target to act upon. Thank you for your kind attention. These are my references.
Thank you very much, Gagan. Judges request you to ask your questions, please.
Thank you, Dr. Gagan. Very, very important study. So I have two queries. One is, what was the clinical correlation? I understand your sample size was about 9 eyes. So what was the clinical correlation? Like, how did you come to the conclusion that, increased expression of the EMT or this is associated with the patient who ultimately developed metastasis or had an aggressive disease?
Okay, so yeah, so I'll take you to the slide before, which I run through because of 6 minutes. This is a heat map. Are you able to see this? Dr. Seema?
I think
it has not shared.
It's not shared.
Just give me a second. I'll share it again because that will answer this question. Is it visible now?
Yes.
Okay, so this is the heat map. If you see on the left-hand side, these are advanced retinoblastoma cases, right? And then these are the non-advanced retinoblastoma cases below. And the dark blues are where you find, if you find the dark blue color in the heat map, that is where it is raised. So if you see in all advanced cases, in this line, in MDR1 here, and ZEB1, you are going to get a dark blue color. So this is where, if you come down to non-advanced, it is going down. So this is the clue there that this is more in an advanced case on this heat map.
Okay, so when you talk about, when you say advanced cases, you are talking about intraocular disease, or you have also included patients who had metastasis?
No, these are all intraocular disease as of now, because these are enucleated eyes. So, and advanced disease, other thing what we are going to say would be this one, if I may again tell, this is a chemo-resistant cell line, topotecan resistant, carboplatin resistant. So these again will be the resistant cell lines where we are getting ZEB1 again overexpressed.
Right. Okay.
I hope I have answered your question.
Yeah, I think probably once it comes out, once you look at the data, like, little more broader.
6 minutes is kind of some data gaps will be there. They are there, but I can, anytime we can discuss over that.
Sure, sure.
Thank you very much, Gagan. Anybody else to ask questions to Gagan? Gagan, can you please share? Yes, please.
It's done, sir.
Thank you.